And here we are picking up from part 1 on themes and control systems in physiology, homeostasis, and graphical interpretation of data, which is the part 2 of the chapter 1 lecture. So to sum it up at this point, we have homeostatic windows. That is to say, we are regulating variables, things like pH or temperature, concentration of sodium, concentration of potassium, blood sugar, etc. We often are trying to control in a range, what we call a homeostatic window. When we say set point or optimal value, that's usually the middle of that potentially optimal range. For instance, we're not always 98.6 degrees Fahrenheit. Maybe you guys are running to class, and by the time you get to class, you're much warmer than you were. Well, your body temperature has risen due to metabolic function. Well, then you may start sweating and so forth in order to get us back to a range, if you will. Let's now move into control systems. Again, this idea of input, integration, output. In physiologic systems, we have two, shall we say, scalable, orders of scale, if you will, of control systems. We have what are called local control, and that's usually either intracellular or within a few cell radii. Let's give you an example. Let's say a mosquito bites you on your arm and you get a little welt there. Well, that's a local phenomenon. The mosquito bit you, and in the process of biting you, you've developed an inflammatory response that occurs right there at the site of the mosquito bite. It doesn't go up to your brain, and then your brain doesn't send a signal down there for that little welt to form. That's happening in the cells and in the tissue right there, what we call a local phenomenon. A reflex, on the other hand, is something where there's a signal sent up to your brain. For instance, let's say that mosquito is biting, and there are some inflammatory signals that then stimulate neurons to go to your brain and slap that mosquito. That's a long distance, what we call a reflex. Both the local and the reflex have the same fundamental pieces of the puzzle. That is to say, there's an input signal, there's some kind of an integrating center, and then an output signal. In the case of local control systems, the integrating center and the outputs are basically at the same location as the input signal. A reflex, they can be widely separated, even extra-organismal in some cases. We'll give you some examples of that. I have a lot of fun with that in the endocrine system. Again, local and reflex. The difference between local and reflex being that reflexes are long distance and utilize nervous signaling or endocrine systems. We will talk about hormones. We'll also talk about a set of diffusible signals known as cytokines. For instance, the fact that you can feel the itching or the burning of a mosquito bite has to do not only with the nervous system sending it up to your brain, but before the nerves are activated, there's actually a little bit of diffusible signal, and that's what we would call an endocrine, a long-distance diffusible signaling pathway, and that's chapter 7 in the next unit. These things are feedback loops. What do we mean by a feedback loop? Is it stabilizing a homeostatic system, or is it pushing us away from a homeostatic window? Let me say that again. Is it bringing us back, stabilizing, if you will? Bringing us back and stabilizing meaning two different ways of saying the same thing. Is it bringing us back to a homeostatic window, or is it pushing us away from that homeostatic window? If it is negative feedback, that's where we are bringing the variable back into a homeostatic window. We are stabilizing it. All right. Body temperature. We get too hot, we sweat, we radiate, we flush in color in order to get that energy out of us, that heat out of us, in order to bring our body temperatures back to a window that we consider a homeostatic set point. So stabilizing or returns us to a homeostatic window. Positive feedback, on the other hand, is reinforcing the departure from the homeostatic set point. Like I said, we breathe normally, right? And occasionally we have to cough, or we have to sneeze, or we laugh and so forth. But let's talk about coughing and sneezing for a moment. The coughing and sneezing are a clear departure from a normal breathing pattern, reinforcing whatever the stimulus is, pushing us away from a homeostatic set point. All right, it is reinforcing whatever is pushing us away from a homeostatic set point. Again, a sneeze is an example, coughing's an example. We will show you examples of positive feedback throughout the semester. We will show you examples of negative feedback throughout the semester. But again, know the difference. Be able to identify biological examples of both. In your book or online, you will see lots of non-biological examples, and they're wonderful, but if you can't apply these concepts to a biological situation, it's not going to serve you very well. So I will require of you that you find biological examples of negative and positive feedback. Feed forward can be either one of the positive or negative feedback situations, but the difference between feed forward and these by themselves is that feed forward anticipates the change before the change actually happens. That is to say, it is a response in anticipation of a homeostatic set point return or depart away from. All right, so we are ahead of the stimulus with a feed forward. Most of the mechanisms that we will talk about with negative and positive are occurring following the stimulus. Feed forward control uses negative and positive responses ahead of the stimulus. All right, that's the big difference. And again, find some biological examples. Here's an example of negative feedback, a very simple phenomenon, body temperature. So take a look at this figure. You can apply it. Again, be able to recognize negative and positive feedback mechanisms in biological systems. And vice versa, we have what's known as positive feedback, where the response to the stimulus pushes the body or the system within the body away from a homeostatic set point, whether that's blood pressure in the case of shock, and other types of situations where it may be beneficial to deviate from a homeostatic set point, but only for a relatively short period of time. And that's why positive feedback is much, much less common than negative feedback, because the negative is always bringing us back to a set point. Positive's pushing us away, and there may be reasons for that. The canonical example, the most common one that you see given for positive feedback, is the cervical stretch mechanism. That is, as the baby gets larger and larger, and especially when it rotates in what's called effacement, that is, effacing the cervix, you begin to get a profound level of cervical stretch. That stretch then triggers a neuronal response that goes to the pituitary gland, actually the brain first, the hypothalamus, which then causes oxytocin to be released from the posterior pituitary. So we get the stretch that then triggers a response. Now that oxytocin doesn't relieve cervical stretch; it actually it enhances it. That's because oxytocin will increase uterine contractions. So now we've got this baby pushing even more against the cervix, which means more oxytocin, which means more contractions, and so forth. If you've ever heard of a baby being induced, That's what they're talking about. What they do is they give the mother some oxytocin. The official name of the drug is called Pitocin. It's pituitary oxytocin. They give this, that which then initiates labor. More push, more cervical stretch, and so forth and so forth. What happens is you have to have an outside factor that then breaks that cycle. In the case of cervical stretch, it's the baby being delivered. Once the baby is outside of the birth canal, then there's no more cervical stretch, and this positive reinforcing feedback cycle is broken. It's not the only example, but it's the most common one that you see. A couple other examples. For instance, blood clotting is a form of positive feedback that requires an outside factor to stop blood from clotting continuously on and on and on. So we're going to be talking about that when we get to chapter 16. If blood is not properly stopped from coagulating, it can form what's called a thrombus. That is a plug of clotted blood, or if that thing breaks off and moves through the bloodstream, they call it an embolus. Another example, just a third example for you, is breastfeeding. So a mother will continue breastfeeding, which is not exactly a what we call a homeostatic beneficial homeostatic mechanism for the mother. She has to expend calories and energy, metabolic energy, and so forth in order to maintain milk production. So as long as the baby is actively seeking and suckling, the mother will continue to produce milk. And this can even occur beyond the first baby. So, for instance, there is a phenomenon or a tradition in many cultures, including American cultures at some time, called a wet nurse. A wet nurse is a lactating female who can then support babies, whether it's their own or others. So as long as that wet nurse remains active, she will continue lactating. Once the baby weans, that is, stops actively breastfeeding for a period of time, that's when the mammary glands will then regress and the mother will stop lactating to a point where even if the baby tries to resume again, let's say a few weeks later, the mother will not produce breast milk. So those are all examples of positive feedback. The key is that you're enhancing the response. So suckling will enhance the lactation response. Cervical stretch will enhance uterine contractions, and blood clotting will actually self-reinforce and trigger more blood clotting. Those are all examples of positive feedback that require some kind of an outside factor, some kind of an outside event in order to break that cycle. In this figure here, what you see are the basic silver thornograms, if you will, of negative feedback. Okay, so we have an initial stimulus. The response causes a diminution, a stabilization of the stimulus. That's why we have the T's here. If it was reinforcing the stimulus, like down here in positive feedback, you'd have an arrow rather than this dashed and or T bar symbol. So negative feedback lowers the amount of stimulus, alleviates the initial stimulus, stabilizes, if you will. Positive feedback causes increased stimulus, increased response, increased stimulus, increased response, and some kind of outside or other factor is required to shut off this reinforcing cycle. There are examples where we don't get that outside factor, and that leads to certain kinds of pathophysiology, things like shock, for instance, where we have anaphylactic shock or some other things. We get a sort of positive feedback cycle, and in that case, the outside factor may be care from a caregiver, for instance. The classic example of feed-forward response is known as the Pavlov's dog experiment, where if a dog will salivate in the presence of food, if you create a signal that could be associated with food, in this case, ringing a bell, if you haven't trained that dog, if that dog has not created an association, then there's no response. But if you deliver food along with the bell, eventually, after conditioning, you will get a physiologic response, even though there is no physiologic stimulus. That is, there's no food present in order to trigger salivation either in the mouth or anywhere in the digestive tract. So, by all reasoning, they should not be salivating and secreting gastric juice. But if there is an association created that would lead to an anticipatory response, that's feed forward. So Pavlov's dogs is a good simple one. One that's every day that we don't really think about all that much are these sort of complex maneuvers that we do, like for instance, walking. So what happens in walking, if you watch the right leg here, it's going to come back. Now, if that right leg is back here and the left leg doesn't swing forward, that person will fall down. And we've all experienced that, accidentally tripping on something where the leg that's swinging forward is not where it's supposed to be, and thus you lose your center of gravity and you might even tumble to the ground. So that swinging forward of the leg is in anticipation of the shift in the center of gravity. So this is walking, this coordinated muscular movement of the legs and in some cases the arms a little bit, is actually a type of feed forward, anticipating the body's shift in center of gravity before it actually occurs. One example of such is, for instance, the volleyball spike, if you will. The person will begin engaging in behavior preparing to strike the ball at just the right time. All right, so this doesn't feel quite as physiologic as other ones, but nevertheless is very, very important for things like walking or other types of complex neuromuscular behavior. Many of those things are feed forward responses where the muscle is tensing up or ramping up. Neuromuscular groups are coordinating with the brain in order to affect complex behavior ahead of when the actual stimulus arrives. In this case, the stimulus is the ball being in the right place there. So we ramp up and undergo this, and this is where you see a lot of feed forward. So if you're seriously interested in kinesiology and athletic training and coaching and so forth, understanding that a lot of the behaviors that you're going to be training your clients, students, and customers is fundamentally about feed forward mechanisms. One of my favorite examples of a feed forward in actual practice is right here. LeBron James continues to try to shoot the ball. If you watch closely here, continues to try to shoot the ball even though the ball isn't even present anymore. You can see that his neuromuscular response is as if the ball is still there and is still moving towards the basket. So this is an example of a feed forward manifestation. The ball's been knocked away, but nevertheless he follows through on the dunk that he was anticipating that he was going to do. You can see this in football where somebody knocks the ball away from the quarterback as they're about to throw it, and they still go through that throwing motion. Again, these complex behaviors, this complex kinesiology, is very many of these things that we observe in sports or in other behaviors. Like I said, walking is an example. You put your foot out in front of your body in anticipation of the body falling forward onto that foot. All right, that's all feed forward. Before we wrap up this chapter, there's one more thing I would like to discuss, and that is every semester, every class, every semester, speaking from experience, a lot of students believe they have a better handle on the interpretation of graphical data than their responses and their ability to articulate that understanding demonstrate. It would be really, really good idea. That's why I say highly, highly recommended that you get as strong as you can at looking at data and and reciting it to yourself in your head. What does this data mean? So oftentimes I'll give data during an exam or something along those lines. And what students will try to do is they'll try to wing it. They'll try to say, oh, well, I can kind of figure this out. Really, it is such a simple question. What does this data mean? What is this data trying to tell me? Why would the authors or the scientists or the instructor spend so much time and effort creating this figure? And what is that figure trying to tell me? They wouldn't have spent that time if they didn't think it was important. So please do take a look at this section. I'm not going to test directly on it, but I guarantee you throughout the semester we're going to have graphically depicted data. Either you're going to have to draw your own or you're going to have to interpret somebody else's figure. And so it would just be a good idea to go over this as part of, just as a double check, if nothing else. You might be a math major for all I know. Well, that's great. Might be a physics major. That's great. Just double check. But if you aren't a math and physics major, it would be a good idea because, again, I see a lot of students leaving points on the table because they don't have the skill for looking at graphic data, and a lot of medical data is graphic. So good luck with that, and if you have any questions, I'll be glad to answer that for you. So to summarize Chapter 1, we're talking about elements of integrative science. That includes things like emergent properties, okay? Function and mechanism, understanding what constitutes a good answer in this class and what does not. Again, we're trying to move you past what a person who's taken a lower course or a person who's a client or a patient. We need to have that context. The themes allow us to see patterns. That is to say, when we are not fully familiar with a given system or response or scenario in the body, how can we use the themes of physiology in order to work out what's really going on? Themes like homeostasis. We know that homeostasis is a goal of the body, but what if the body is not responding in the way that we would have anticipated based on what we would consider a homeostatic set point? Then we start to look at control systems, negative, positive feedback, feedforward, in signaling and information flow, receptors, integrators, local, reflex. It's surprising how much information is in Chapter 1, given that it's such a general chapter. So please, even though it's a sort of a hand-waving chapter in many ways, use this chapter as a toolbox for working through the rest of the chapters in Unit 1. With regard to Chapter 2, there is no assigned reading for Chapter 2. But here's what there is a mastering A&P assignment online for Chapter 2. If you've had trouble with chemistry and or Bio 165, this chapter could be a real lifesaver for you. And it would be worth just taking a little bit of time looking at the highlights of the chapter. Maybe identify something that you're not as strong with, like for instance pH or ions or those sorts of things. Would be a great review for you. Again, you'll have an assignment, but no reading. So this concludes our Chapter 1 material. As always, if you have any questions, please reach out to me, and I'll be happy to answer any questions that you have. And we'll talk to you again soon. Thanks.